Now we are going to move on to eukaryotes. We probably won't be able to finish this part, but we'll get started on it. In prokaryotes, they're very, very diverse. There's bacteria and there's archaea. Notice that archaea is close to eukarya. That means it's more closely related to eukarya in some things, but because they don't have a nucleus, it's a prokaryote. So prokaryotes came around first, then it got more complex and turned into eukaryotes. In domain eukarya, we have protista, animals, fungus, and plants. In this chapter, we will go over protista first and then fungus. Chapter 16 is plants. Chapter 17 is animals. So we're going to go over in chapter 16 and 17, we're going to take different type of plants. We're going to group them together. Same thing for animals. For example, fish, which group they go to, mammals, amphibians. So we're going to start doing that in chapter 16 and 17. Today, we're going to cover protista. So what is protista? If it's an organism that is not an animal or fungus or plant, it's protista. Protista is very diverse group. They do some DNA studies to see if it's closely related to plants, fungi, or animals. If it's not, then they put it in this group called protista. So that's the first group we're going to cover. So here, so we did prokaryote, now we're doing eukarya. And under eukarya, we have four groups. We have protista. There is fungi. Those are the two we're going to cover in this chapter. Plants and animals. That's chapter 17. It's protista if it doesn't fit into all these three other groups. So then it's protista. Let's look at protista. Actually, before we even do that, let's talk about where the eukaryotic cells came from. So we already kind of talked about the Miller experiment, how organic molecules came around. Then there was lipid membranes and turned into protocell, first cell. Then that got a little bit more complex, turned into prokaryotes. Then these prokaryotic cells, they got even more complex. They turned into eukaryotic cell. Eukaryotic cell has a nucleus, and it has other membrane-bound organelles like mitochondria, endoplasmic reticulum, smooth plasmic reticulum, Golgi apparatus. Those are all the organelles. We covered this, I can't remember the chapter, but it was called endomembrane system. So it was endomembrane system. There's endo means inside, membranes. A membrane is referring to membranes, so there are a lot of membranes inside. So what is the origin of these organelles? So let's look at that, how we might have gotten these cells from prokaryotes. So this is the first cell, protocell. You have the membrane and you have some genetic material inside. Very simple. Then over time, this membrane started to fold. So membrane enfolding started. And then it got a little bit more complex, and there were lots of internal membranes, and there was a membrane that formed around the center. That's how you got the nucleus. This is also made up of phospholipids, this membrane. The whole purpose of this was to increase surface area. So things that go in, if there's more space for them. So to increase the surface area. So you started with a simple cell, protocell, that converted into prokaryotes. Then the membrane started to unfold, and then you got more organelles and also a membrane around the nucleus and around the genetic material. So there is something called endosymbiotic theory. So it explains how these eukaryotic cells got mitochondria and chloroplast. Mitochondria and chloroplast, they're powerhouse of the cell. They help creating ATP. So the endosymbiosis theory proposes that mitochondria and chloroplast were their own organism, free living bacteria that got eaten up by other cells. So here is the common ancestor, the protocell, with the genetic material. Different types of this protocells. You have archaea, you have some aerobic bacteria, that's mitochondria. You have some photosynthetic bacteria, some chloroplast or chlorophyll. So this membrane enfolding started, and it started engulfing these bacteria. But because this bacteria is very efficient in producing ATP, it said, let's make a deal here. Let's do a bargain. I will give you a nice, safe place to live. In a return, you can give me ATP. So that's how the organelle mitochondria came around in eukaryotic organisms. It got engulfed, and then now they're living in mutualistic relationship. Mitochondria is getting space to live. Eukaryotic organism is getting ATP in a return. Indo means inside. So indo means inside. And symbiosis means living together. So that's how they started living together, mitochondria and eukaryotic cells. Then there was photosynthetic bacteria that was also its own bacterium. It also got engulfed by this bigger cell, and this also gives the ability to make ATP. So plant cells, they have mitochondria and chloroplast both. Animal cells, they only have mitochondria. You only have mitochondria. So they were their own cells. They got engulfed and they started living in a symbiotic relationship with these eukaryotic cells. So mitochondria originated as aerobic bacteria, chloroplast originated as photosynthetic bacteria, and genes moved from the DNA of new organelles to the nuclei of host cells. So these got engulfed, and then they lost the ability to live on their own. So what I'm trying to say is today, because they are in a symbiotic relationship, if we take away this mitochondria from this eukaryotic cell, it wouldn't be able to live, or chloroplast. So now they're codependent on each other. They cannot live on their own any longer. So again, just to remind you, plants, they have mitochondria and chloroplast both. This mitochondria have its own DNA. So there's, so they use this to explain this endosymbiotic theory because it has its own DNA. They say it was its own cell at one point, own bacterium. Also, if you look at the size, they're really small in size. So that's another thing that supports endosymbiotic theory. So they have their own DNA. That's what supports it. And they also have, they're also small in size. So that's endosymbiotic theory. Make sure you remember it because I might ask you how the eukaryotic cells got these two organelles, chloroplast and mitochondria. Then you have to tell me through endosymbiosis or endosymbiotic theory supports that. So that what we just went over was primary endosymbiosis, means early eukaryotic cell engulfs aerobic bacterium that becomes mitochondria, also cyanobacterium that becomes chloroplast. That gives you early green algae. That's what produced oxygen and caused the first mass extinction. Then there was something called secondary endosymbiosis, mean this happened twice. So in this, you have this eukaryotic cell that engulfed this early green algae. And now it has both. It has chloroplast and it has mitochondria, and the nucleus has the genetic material. So that's the secondary symbiosis. So some eukaryotics, they have chloroplast. We call them plants. With the three membranes. So they can see that there are three membranes, and that's how that happened. Three membranes came from primary endosymbiosis and secondary endosymbiosis together. So there has to be two endosymbiosis events for it to have three membranes. So that's what supports that when they look at the membrane system. So that's endosymbiosis. There's a question, which of the following lines of evidence would not be consistent with endosymbiosis theory? A, chloroplasts have the same photosynthetic pigments as cyanobacteria. Mitochondria have their own ribosomes. Chloroplasts have their own DNA. Mitochondria have their own nucleus. Mitochondria and chloroplasts have their own DNA. That's what supports the endosymbiosis theory. So most of you said D. That is true. This is the answer. Mitochondria have their own nucleus. This is not true. So mitochondria have their own DNA, mitochondria and chloroplast, but they do not have their own nucleus. Also, mitochondria have ribosomes, so they have their genetic material. That's ribosomes. And then chloroplasts have the photosynthetic pigments as cyanobacteria. So that's another thing that supports the endosymbiosis theory. Okay, so let's see. So we have few minutes. We have 5 minutes left. Let me just go over this slide. So we just talked about eukaryotic organism. Now we're gonna talk about how those eukaryotic organism turned into multicellular organisms. We have a jar of this in the lab. This is called Volvox. And this is a between a group of individuals and multicellular organisms. This is kind of like a bridge between single cell and multicellular organism. All these cells are a lot of cells around there, and these are the reproductive structures. I need you to know the name of this. So if you were in the lab today, you would be taking some pond water, putting it on a slide and putting a wet mount, covering it with a cover slip, and putting it under the microscope and looking at this. So this is called Volvox. This is the bridge between basically multicellular and single cell organisms. That's Volvox. So I'm going to give you some names today that I want you to remember for the exam. So now we're going to start getting into Protista. So Protista it's going to have organisms that do not fit under animals, fungus, or plants. That's Protista. So that's the first thing we're going to go over. After that, we have fungus. So kingdom Protista contains eukaryotes that do not fit into any of the other three kingdoms. So in animals, fungi, or plants. So these are the kingdoms. Just to kind of remind you, these are the kingdoms. This is the domain. So I am going to stop here, and next lecture we will go over Protista and then fungus, and we might even start plants chapter. So I'm gonna stop here.